evening, everyone. My name is Sarah Howard, and I'm the Youth and Community Manager here at the Danube Regional Library. And we are happy to have Science on Wheels through MU here with us this evening with several scientists and many different topics to share. So please do feel free to put questions in the chat, and they'll be able to answer those after each of their presentations. And it's my pleasure to turn this evening's introductions over to Zach Miller, part of the executive team for Science on Wheels. Have fun.
Hello, everyone. My name is Zach Miller, and I am a PhD candidate at the University of Missouri. And like Sarah said, I'm one of the executive members of Science on Wheels. And Science on Wheels is an organization run by graduate students like me and like everybody else that you will see here tonight. There's several people from our exec team. And our mission is to just to share some of our science and also just to connect with the public of Missouri. And we have scientists from different departments across engineering, across all of the STEM categories, studying a wide range of material from trying to figure out cures for cancer to understanding the evolution of miniaturization in lizards to agricultural plant biology. And we will be traveling, we'll be back to doing in-person stuff, I guess, at the library next spring. And this is our third event held virtually. And thank you to Sarah for hosting us again. And without further ado, I will introduce our first speaker tonight, who is Katie Horton. And she is a second-year PhD student in the Department of Plant Biology. Take it away, Katie.
Thank you, Zach. I am very excited to be here tonight. So, all right, I'd like to talk a little bit first about myself. Everybody loves talking about themselves. I'm from Northeast Georgia. I got my undergraduate degree in biology, and I did a lot of work as a field biologist and botanist, just kind of in general. I've got a couple other humble brags here, but we don't need to go into them. My undergraduate research focused mainly on plant phylogenetics and plants that grow in weird special places. This is a greasy grass. Don't know why it's greasy. This is a sweet shrub that grows on the East Coast. It probably has some cryptic species in there. And this is a butterfly pea. This is the one that grows in most of the Southeast, and this is the one that grows only in the Florida Keys in special rock. So that's where I came from. And now I'm in Missouri at Mizzou. And I want to talk about the research that I'm doing here. So I specifically study plant immunity response and how plants respond to pathogens that are attacking them. And the way they do that is they recognize these patterns that all pathogens have they can't live without. So that'd be like recognizing a person's heartbeat. So like I see your heartbeat or I hear your heartbeat, so I know that you're there. That's how the plant recognizes the pathogen. Obviously, that's not good for the pathogen because it can't just get rid of whatever that is. You can't get rid of your heart. So the pathogen makes these things called effectors. They're little proteins, and all you need to know is the effectors come over here and they stop the plant from recognizing those patterns. They stop the heartbeat from being recognized. So now the plant's unhappy, the plant's getting sick. The plant makes some proteins that can recognize these effectors, and this is as simple as it can get for this interaction. It gets a lot more complicated, but basically all you have to do is think about it as the effector and the resistance proteins fit together like puzzle pieces. So we have the ABR-RPS4 effector puzzle piece over here, which is the effector that I study. And then over here on the right side, we have RPS4 or RRS1. And those are the resistance proteins that specifically recognize that effector puzzle piece right there. So the plant makes these, the bacteria makes these. It is round and round and round with the trying to adapt to avoid disease here. So we found some previous research in my lab about the way that certain plants responded to this. We look at lettuce and we also look at turnip, which are kind of far apart evolutionarily, but we found out that they respond to this specific effector differently. They recognize it differently. Their puzzle pieces fit together in a different way. And this slide is complicated, but all I want you to see is that this is the whole effector. This side of it is green and this side of it is orange. It has two pieces. When it goes into the plant, that breaks into two separate pieces, kind of like if you were going to take an Oreo and split it into the two separate ends and then eat them separately, because that's how Oreos are meant to be, I'm pretty sure. So this is what it looks like when you put this effector in lettuce. Let's call it the left side of it with the in over here, does not have any disease response. The right side of it, the left side of it does have a response. The right side of it does not. And the whole thing altogether is kind of uncertain. Our model organism, Arabidopsis, is kind of in between. And then turnip does it entirely the opposite of lettuce. Turnip says, I don't care about the left one, but I do care about the right one. And what's interesting is that RRS1 and RPS4 are supposed to be required for the recognition of that right side of the effector, that part of the puzzle piece. But there is no known RRS1 in turnips. So what am I doing about that? I'm growing a lot of turnips. I'm going to take that effector and I'm going to put it into the leaves of all of these turnips and see if I can find any that respond in a way that will allow me to figure out what is recognizing that effector. Why is it responding that way? Where is the other puzzle piece? So if anybody has any questions about that, I'd love to answer them. This is a little plant that is related to turnip and broccoli and all of those. It grows in Missouri. I found this out in the Gans Creek wilderness area. So thought it was cute, thought you'd like to see it. Lots of questions. Thank you all so much. What got me interested in plant diseases is the first question. And so that's actually a really fun thing to answer for me. I have always wanted to work with plants in space, and plants in space tend to get a lot more diseases than they do on Earth, which wild, I know, because you think space seems like such a sterile environment, but really it's just anything that can eat something else wants to. It's stressed out up there. So it's the first step, really. The next question is, how do you find an effector in a plant? That is the question, isn't it? So these puzzle pieces are numerous. And so the effectors are coming from the bacteria, and we're just putting the effector in with like a syringe right now, basically. But you could isolate it from a plant by doing this thing where you can pull down all the proteins and look at how big they are, or you can, if you wanted to find the resistance gene, what we're planning on doing is comparing the genetics, comparing the DNA of all of the plants that do respond and all of the plants that don't respond. So it's kind of a big project. Next question, what is your favorite plant disease in either Arabidopsis or turnips? My favorite plant disease is the one that I intend to be there. Gosh, otherwise, I don't know. I don't know if it counts as a disease necessarily, but I really like parasitic plants. I think they're super cool. It's kind of a disease. Parasites, Thank you all for the questions.
Okay, thank you so much, Katie. That was a great presentation. And next up, we have Frances Di Donato from the Department of Natural Resources, who's going to tell us about her research on geese. Take it away, Fran.
Hey, thank you. Okay, so I don't really have much of a PowerPoint set up, but I do have just some pictures that I want to have. So I'm going to share my screen now and see how that goes. Let's see. Oops, sorry. Okay. To presenter mode. Okay, sorry about that. All right, so everyone can hear me okay? That's on the panel at least?
Yep,
Yes.
we can
Okay.
hear you.
Okay, and you see the picture that I have up as well?
We do.
Okay, great. So thank you for the introduction. My name is Fran Di Donato, and I am a grad student in the Natural Resources Department at Mizzou, and I'm studying waterfowl. I'm originally from New York, and I've always been really passionate about the outdoors and wildlife, which led me to pursuing this as my career path. So today I'm going to be talking to you about my research on geese, but not just any goose. I study a specific type that spends the summer way up north, raising their young in the Arctic, and then migrates to spend winter on the Atlantic coast. And even more unique is that these geese actually prefer to concentrate during winter in the concrete jungle that's New York City, Long Island, and the Jersey Shore. These geese are called Atlantic brant, and they look pretty similar to Canada geese, which are probably very familiar to everyone in Missouri. And so that's what I have here on the picture is the Atlantic brant here, and you can see how it's similar color scheme to Canada geese with the black head, some white on the neck, and just sort of an overall grayish brown body. But brant are actually a lot smaller than Canada geese. In fact, they are only a little bit bigger than ducks. I have another picture here just showing the size comparison between the brant goose and an American black duck, which is just a specific type of duck. But you can see they're pretty much the same size, but they just look slightly different in their morphology and the shape of their bill and their body size. And the bill, that's their beak. And so also about brant, their population is pretty small. We estimate there's only about 150,000 of these birds. And compared to other duck and goose populations, which may number in the millions, it's really important that we closely monitor the brant since there are relatively very few of them compared to other waterfowl. And today, I'm going to talk about how we study these brant geese in the urban jungle of the greater New York City area and the Jersey Shore. So one of the things we're interested in is we want to learn more about their survival and their social behavior, as well as their reproduction. So we started a program where we catch brant and we tag them with a colored plastic band that has a unique letter or number combination on their legs. And then we release them back into the wild after that, completely unharmed. I have another picture of these color bands that we put on their legs. You can see this one has a 9 on the left leg and then a P on the right leg. So when we catch these birds, we actually use rocket nets and we set them up in soccer and baseball fields, public parks with grassy lawns, and especially at golf courses because the brant love to eat grass. So that's where you find them most in New York City, is on any of the golf courses or any fields around there. So rocket nets are propelled by explosive charges And what we do is we fire the net over the brant once they get close enough to it. And the net, it just lands over them, and we quickly remove the birds so no harm is done. And then we put these plastic bands onto their legs, record any other information like their weight, their sex, different measurements, and then we set them loose. So after this, after they've been marked with these colorful plastic bands that are unique to every bird, we can go out and find them again. And what we do is we develop a tracking history for birds based on their observations. So a big part of my research and the field work that I do is going out and trying to recite these color banded brant. So I travel all along the Jersey Shore, New York City, and Long Island during winter, and I visit just about every park, school ball field, golf course, any green space, just looking for brant. And when I find a group of brant, I set up what we call a spotting scope, and it just looks like a telescope that I set up, and it functions similar to binoculars, but just has stronger magnification. And I will scan through the flock of birds, focus on their legs to see if any of them have these bands, and then I just do my best to read the combination and record what I see. So I think I'm going to stop sharing this picture. I think that might make my icon bigger, but I want to show how big the plastic bands are relative to me, I guess, because it's hard to judge. But so this is an example of the band. So if you see it up against my hand, and just imagine that's on the leg of a goose way off in the distance, and it's running around. So that's why I have to use such a high-powered scope to try and read these little bands. So it can be pretty hard, and the brant also like to congregate in groups, sometimes more than 1,000 birds. And one of the things that can also make it difficult is that sometimes there are people actively golfing on the courses where the brant are frequenting, as surprising as that is. So being in such an urban environment, the brant have grown pretty comfortable with humans, and they'll let you get very close to them. But sometimes the people do disturb them, so they'll just get up and fly around on the golf course, and I basically just follow them around. So one of the stories that I have from conducting this research is in such an urban area is that I stumbled on a group of brant that was in a soccer field, and it was just next to a recreation building, and it was nice weather that day. So the rec center just decided to have an outdoor aerobics class, but the brant didn't seem to mind and just carried along. So I was out there collecting my field data as an aerobics class was just going on. And so I've had lots of experiences like this where the geese are in close proximity to the public in the urban environment. So as I mentioned before, with these recitings, we document unique birds and we record their known observation history. And this data is what we use to estimate their survival probability. So we use the amount of times that they were observed over many years, and then the time spanning in between each of these observations to develop a probability of reciting it. And then that is incorporated to estimate the probability that they survive or were they just missed in that time frame. And so that's what we're doing with this information. So we've estimated from some of our work that brant survival is pretty high. It's estimated about 80%. And we have records of brant living upwards of 15 years in the wild. So if you remember from earlier in the talk, I mentioned that brant migrate to and from the Arctic back to New York City every year, which are over 1,000 miles between each other. So imagine making that flight 15 times. I just wanted to end with one little story about some of my field experiences researching the geese. Because I do spend many weeks observing the brant during winter, I start to get to know some of them and their unique codes if I continue to see them throughout the season. So my favorite birds are named 4T-BB and 5T-BB, which just, it reflects the color and letter combination on their legs. And I first documented these two brant together in November of 2019 on Long Island. And I determined that they were actually a mated pair and they had one gosling in their family that year. And I continued to see them throughout that winter with their one baby, and they stayed together as a family throughout the whole winter. And every time I would notice one of the birds, I knew that the other two were closely behind, and I would see them just in a few moments. And what was most exciting was that I saw 4T-BB and 5T-BB again in November 2020. So that means they went back up to the Arctic and came back down to New York City again successfully. And I saw them all throughout last winter. So I'm really looking forward to this winter to see if 4T-BB and 5T-BB come back and are back on Long Island, but we'll have to wait and see. So we still have a lot to learn about Atlantic brant, just with this color banding program and recitings just beginning, but we're on the right track to learning more and helping better understand and protect this species in the future. So with that, I will take any questions. Okay. Let's see. Okay, so the first question is, do the color bands affect the way they pick mates at all? And what is the best way for you to see a brant? And if you do, is there a way that you can record your observation? So we don't believe that there is any differences in the pairing and finding mates based on the plastic bands, but it's difficult to tell because we don't have any controls because we can't identify individuals that don't have the bands. So we don't know much about their pairing unless they're already uniquely identified. So that is an interesting question that could definitely use some further research. And then the best way to see a brant, really, they are extremely common and very easily observable if you visit pretty much anywhere along the Jersey Shore, or even in New York City, just on the southern shore of Long Island. In New York City, they can be seen in big rafts, big groups out in the bays themselves, but they will come onto the land, and they get into groups of thousands onto the golf courses. So if you go to a golf course in New York City in winter, or anywhere along the coast of New Jersey, you might be able to find them. And these bands are reportable to the Bird Banding Lab, which is based out of Maryland, and it's coordinated through the U.S. Fish and Wildlife Service. So you can technically report those to, I believe it's called reportband.gov. I'll put that in the chat, reportband.gov. Or I should also put my contact information because you can report these observations to me directly. So put my email in the chat. Okay. Let's see. Okay. How dangerous is it to manipulate the birds when you capture them? It's not very dangerous. They are pretty calm once you get them, and their bills are kind of flexible, so they can bite you a little bit, but it's really not very dangerous once you have them in hand. And then another question, what are the pros and cons of marking recapture versus something like telemetry and other tracking devices? So I would say one pro that is very obvious is that some of those devices can be really expensive just because of the data that they collect. So they are able to collect a lot more finer scale information than these observations, but some of those transmitters and devices can cost thousands of dollars each, and these plastic bands just cost only a couple dollars. But we don't get the same amount of information back just because we have to release a lot more bands and then go out and find only some of them, whereas with these transmitters you might be able to find more, but you would only be able to put out a few, but you get most of that data back. And so I think it also just depends on the research question that you're asking too, because these tracking devices could be a lot better at answering fine scale habitat use and movement. And these color bands, marking and reciting are for more broader scale patterns, like estimating survival across a population. You have to wear specific clothing to protect from the winter while branch searching. It can get very, very windy along the shore during winter times. So I do my best to keep most of my skin covered up. I usually wear face masks, hats, and gloves because the wind can just cut right through you. So windproof clothing. And actually, because brant are waterfowl, they do really, their activity increases when it rains. So some of the harshest conditions I have to endure are cold and rainy days during winter. So I do have to wear rain gear because the brant are still out and active in the rain, if not more active than they are on nicer weather days. Are brant as mean as the Canada goose? I would say they are a lot more docile than Canada geese because the Canada geese are so large that they do fiercely protect their families and their territories, especially during spring. And the brant don't seem to do that so much. So I haven't heard of many people getting harassed by brant like they might by Canada geese. And do brant geese typically mate for life, as 4T and 5T BB appear to have done? We do believe that brant do mate for life, but they most likely will replace their mate if one dies. But they do establish this strong pair bond because we believe that it ultimately helps them to be more successful in raising their young, because it takes a lot of energy and effort to find a new mate every year when you go back, when you're flying all the way back up to the Arctic. And so it's more beneficial for them to stick with the mate through that, is the theory. And I believe that's all of the questions, and I'm sorry for going so over time, but thank you so much for the great questions. And thank you for having me here today.
Okay, thank you so much, Fran, and great talk. Next up on the list, we have Alejandro, who is a first-year PhD student in the Department of Natural Resources here at Mizzou, and who will tell us a little bit about his research studying birds or bison or something else cool. Take it away, Alex.
Hey guys, I want to tell you a story. This is that's what I'm gonna do tonight. So let's go back in time. Let's travel 200 years ago. It's the year of 1821. We are somewhere around here, not very far away from Columbia. So 1821, Missouri just turned the 24th state in the US. And as far as we can see, there's this beautiful and vast grasslands. And actually, in the entire North America, grasslands are just vast and uninterrupted extensions of land going all the way from Canada to Mexico and from the Rocky Mountains to Illinois. But that was in the past. Slowly, or what appeared to be slowly, the landscape changed. And it was actually an incredibly fast transformation. So if we go on into the future, but still the past, in the 1872, two important things happened in North America. The first thing is that Yellowstone became the first national park in the US. So that's happy, right? That's cool. But the second news back in the day was that the military allowed the extermination of the American bison, allowed it and encouraged people to hunt down bison as a plan to starve Native Americans. So an average of 5,000 bison were killed every day for about 30 years. And by the end of the century, which happened 28 years after this, since this extermination started to happen, like 30 years after, only 750 bison remain out of the 7.5 million bison that were estimated to roam North America. Most of these remaining bison were in zoos in Canada and in Yellowstone National Park. In about 100 years of constant effort to preserve the bison, we achieved the unimaginable and we brought back those numbers up. So we went from 750 individuals to what we have right now, about 400,000 bison. So that's a clear example of how conservation works, and it's a good example of conservation success. So we solved that problem. Bison are good nowadays, more or less, but we have another problem. It's probably bigger than that one. So, and it's the shrink of the grassland habitat, the grassland ecosystem. So over 200 years, we have lost about 80% of the grasslands in North America, and only a few patches remain scattered through North America. And with that, a lot of the species related to this ecosystem are declining very fast, especially grassland birds. So in 60 years, only 60 years, just think about it. Since 60 years, like your parents were probably around here, right? They were babies. So in 60 years, we have lost about 70% of all the grassland birds in North America than we have. That's scary, right? And this is linked to the loss of habitat, the loss of the grassland habitat. So just like the bison, can we bring back not just a species, but the entire ecosystem, the entire grassland ecosystem back? And how do we do that? So this is where grassland restoration comes to play. Restoration is a direct management action that we do to land to bring back the elements of the ecosystem that got lost during human development. Things like prescribed fires, reseeding, reintroduction of bison, and even like management of livestock, recreate the condition of grasslands before the transformations that we people did. So, but restoration is challenging, and it's not as easy as it sounds like. Restored grasslands show that they support less plant species, like less diversity of plant species, and hence less species of all like animals, like grassland birds. So just like restored grasslands are not just like, they're very different than grasslands. They're not quite the same. It's better than what we had before, but it's not the same. So how do we get better grasslands? How do we like get this grassland back? My name is Alejandro Chávez Treviño. I'm a first year PhD student at Mizzou, and my research focuses on spoiler birds, bison, and grassland restoration. So my research focuses on how different restoration strategies affect grassland birds and grasslands, right? So specifically the probability of a nest to be successful. So many nests during the breeding season fail, or they get predated or something, and this is directly related to habitat conditions. So I spend most of my time in the summer looking for nests and estimating these probabilities of success. So the idea is that a better breeding success is related to a better grassland condition. Another way to know what good habitat is, or what a good restoration is looking at the habitat selection of some grassland specialists, birds, right? So what we do is we catch some meadowlarks, which is a prairie bird, and we put them, we give them a little backpack, and this backpack is a GPS transmitter. So it will take locations of where the bird is spending most of its time during the summer. So the good habitat is where the bird spends most of its time, and bad habitat is where the birds avoid, so which places this bird is avoiding, right? And the third part of my research is about the mighty bison. So I am looking at how like different grassland restoration affects grazing patterns of bison, and how these grazing patterns create different habitats for other species. Remembering that bison were a very, or there still are, very important piece of the ecosystem, since they change the ecosystem and provide a habitat for different species. So that's what I do. Today, there's an increasing number of grassland restoration projects in North America. And according to a study from 2014, the rate of restoration finally has outweighed the rate of degradation. So, but there's still a lot of things that we don't know, and there's a lot of things to discover to restore our ecosystems in the best way possible. And we are looking at that. So I want to finish my talk just with a few words. I want to say that extinction is forever, but extinction is also optional. So let's preserve our wildlife ecosystems, and so that in future, in like 100 years, our great grandchildren can tell the story of how we brought back not just one species, but an entire ecosystem back. Thank you. Yeah, questions. People have questions? Let's see. Let's do this. Okay, here. Why don't restore grasslands support as much wildlife as they should? That's a very important and difficult question to answer. But basically, no, it's legit. It's like a real good question. Basically, there's an ecosystem. It's a very complex network of processes that are happening. And once all of these processes are disrupted, like putting back all of the pieces together is very difficult. And then we can try to recreate those conditions, but they seem not to stick because something is missing. And maybe it's just time, maybe it's something else. So it's like trying to recreate a very complex like machinery that we don't even know how it works, and we're just trying to put it back together. So yeah, that's how I see it. Like imagine that you encounter like a very complex like machine that you don't know how it works, and it's like, hey, how do we make it work again? People is working in that. So where are the main areas you study bison in the US? Oh yes, that's a good question. Like the location? My study site is actually close to Chicago. It's a preserve an hour southwest from Chicago, and it's called Midway National Tallgrass Prairie. I'm gonna write down here, Midway National Tallgrass Prairie. And it's really interesting because it's really close to the city, but I mean, relatively close. But yeah, it has a herd of about 60 bison, and they're roaming around, and they actually are actively trying to restore the tallgrass prairie there. And they have they have been working for a long time there. And this place used to be a artillery factory that they used like make bombs and stuff like that. But now it's no longer that. Now it's a preserve. So what is the minimum size of a prairie restoration that can support bison? Big. I don't know the exact the exact number of acres, but they require a lot of infrastructure because they're not cattle. They are bigger, wilder, and you require like very strong fences and a bunch of brave cowboys to manage them. So big. I don't know. Do you study other birds in addition to meadowlarks, and do you have a favorite? Yes, I actually study the whole community. Another part of my study is to see how like an entire community of birds changes across the restoration process. So so all of them, pretty much all of the grassland birds, I'm into them, but my favorite, I think, is here in the tallgrass prairie. I think it's a Henslow sparrow. Henslow sparrow. But my favorite of the grasslands is the American kestrel. It's like a little tiny falcon that is really cute. How much do the bird backpack weight in relation to the bison color? That's a question that I know because I was just looking at this a couple days ago, and it's 3.5 grams. And the bison color, it's, I don't know, like a kilogram, like 2 or 3 pounds. It's huge. Actually, I think I have some pictures, but I don't have them like with me right now. Where all do you think agriculture plays in restoration? That's a very interesting question, because one part of a restoration is growing soybean. And then once you have that, you take out the soybean, and then you do like, you burn it, and then it's like an entire process, but yeah, that's like a part of restoration. Do you know if the US and Canada work together in preservation of North America? I know Alberta is part of the bison restoration. Yes, and actually Mexico too. So there's this some, how is it called? Well, for birds, there's this project that is a tri-national project that they are trying to look at grassland priority conservation areas across Canada, the US, and Mexico, and they all do like a lot of work with landowners and conservation and bird conservation in particular. So yeah, we all work together. And that's about it. If there's no questions, I'm gonna pass it to the next person. Thank you very much. Appreciate it.
All right, thank you very much, Alex. Okay, next on the list we have Zachary Traylor, who is a first-year PhD student in the Division of Biological Sciences, and will share a little bit about his upcoming research plans studying corn.
Yeah, thanks, Zach. So, like you said, I study corn, and so I think when most people think of corn, they think of the large yellow, kind of plain colored corn, but something that I think a lot of people don't know is that corn comes in all colors of the rainbow. There are different varieties that can be expressed in all various colors, designs, shapes, patterns, and so I have one. I don't know if it's a little easy, but it is purple, blue, white, little speckles, little stripes. They're a very diverse group of plants. And so I study those different varieties and the ways that we can use them, because right now the yellow corn that we all eat, use for grain, all of that is kind of just a monoculture, or it's just one type, one kind of group of corn, and there's not a lot of diversity. And so I want to look at corn diversity, try to expand our usage of these different corn varieties, and kind of fill in kind of niches where different varieties could have better applications. And so in part of that, there's a whole range of things you can look at in corn to determine what makes, this line a red corn special or different from a blue corn. Specifically for me, in my lab, I look at sugar and the different kinds of sugars that are made. And so we all know that corn, being a plant, takes its energy from the sun, uses photosynthesis, and turns it into sugar. And that sugar is converted into energy to make the plant grow and be happy. But I'm studying the ways that maybe these different lines make different sugars and how they are accumulated or stored in the kernel, and what applications in food they could have. And so these different sugars can propose different metabolites. Those are the things that are broken down to make energy. And so I can look at how they are produced in corn, how they are transported from your photosynthetic leaves that are called source tissue, down through the veins, down through the xylem and the phloem, to the kernels, which is the sink tissue. And so I can kind of understand every kind of step of the plant's life cycle, see where different sugars appear, and from there kind of analyze those different contents. And so to do that, I take a nice ear like this, I grind it up into a fine powder, and then I can cook it or process it in water with some enzymes, break it down into just sugars, and then I can use a really advanced machine. It's called a high performance liquid chromatography machine. It's a real big word, to analyze the different sugars, and it just breaks down, gives you a whole list of just the different sugars that are contained in my kernel. And so I can see what's different, what's unique, what's cool, interesting, and obviously, do they taste good? And so hopefully kind of the end is, like I said, we'll make a diverse range of corn. We will fill them into different roles. I know kind of one of the projects we're looking into is making, corn chips out of them or tortillas, to kind of make a new diverse range of corn and food products. And so, it is key to the, I think, diversity and growth of our agricultural requirements as a human race. And so with that, I'll take any questions. I know I kind of keep it short, but I do enjoy more feedback and conversation. My favorite corn color, I love bronze. It's kind of like a nice orange glow, but they really have like a really nice shine to them. And I really love how they look in the sun. They look like your perfect popcorn kernels, and they're really, really good. Let's see, what is the best corn that I've tried? I have tried a white sweet corn that is actually native to Missouri. A lot of my corn that I'll be working on are native to Missouri and Missouri only. And so even in Missouri, we have a wide range of those colors and shapes and sizes. And so we can get a lot of things from our heritage corn. And so my favorite one that I've tried is a white sweet corn, I think from southwestern Missouri. And it's really delicious, especially in popcorn. Is there a way to get seeds for heritage corn? I know we source those specifically from USDA. I kind of have my co-advisor, Dr. Sherry Flint Garcia. She works for USDA, and she sourced all of those seeds for heritage corns from the USDA Seed Savers program. And so I think they are in limited distribution supply, but you can reach out to them to kind of find them. Have I had blue corn tacos or Mexican food? Yeah, I've had blue corn tacos. I think they are delicious. I think they are honestly way better than the yellow corn tortillas or just regular corn chips. And so 100% support blue corn chips, blue corn tortillas. What is the most unique corn variety I know of? That is a tough one. They come in whole ranges of, like I said, shapes, sizes, colors, patterns. I think one of the most fun one is just it's called the Bloody Butcher. It is dark red as you could get, and it is kind of almost a scary looking thing of corn, but I think that one's pretty unique and fun. What has been the sweetest, highest sugar content corn you've tested? Right now, I have not actually been able to test the sugar contents of those corn lines. I'm kind of working on them. That's, like I said, Zach Miller kind of specified that it's in progress. Obviously, research takes money, and so we're kind of working to get the money to do that. And hopefully, by the end of this year, early next year, I'll be able to tell you what is the highest sugar content in corn I've found. What is the part of your project in progress that you're most excited about? I really love the idea of looking at what unique sugars are found in corn and what makes them so different. Like I said, I've tried many of these different varieties. They are all delicious and they are all unique. And so I would really love to know exactly what that sugar, what that metabolite is that makes it special, and kind of how we can take it. And then finally, let's see, what else? What's your favorite corn recipe? One of the people I'm working with in my project, Gary Heingardner, he is another corn user, and so he makes the best blue cornbread you've ever had in your entire life. It is delicious. He doesn't share the recipe, it's a family secret of his, but maybe one day I'll try to get it from him as a celebration for the hopefully end of my project. And then, can all of these sugars be found in teosinte? So teosinte, for people, just so they don't know, is the old version of corn. Teosinte is what the indigenous people of South America first cultivated. It was a little bushy grass that they saw had some good seeds. They've cultivated over the years, and teosinte is what corn, or what has become modern day corn. And so a lot of the sugars we found kind of differ. And so I will also be kind of looking into teosinte, seeing what that line is. And so we're kind of also working on that and developing what those different ideas are. And is there a difference between corn and maize? No, maize is just known as by its scientific name as Zea maize. And so we just call it maize, but corn is Zea maize, subspecies maize. Any other questions?
Awesome. Thank you so much, Zach. Great talk. Next up on the list, we have Ryan Swanson, who's also a first-year PhD student in the Division of Biological Sciences, and will tell us a little bit about his work studying plant gene editing with viruses. Take it away, Ryan.
Cool. So, hello, everyone. Like he said, I'm a first-year PhD student, still doing rotations and finding the lab that's the right fit for me. But today I'm going to talk about some of the work that I did while I was an undergraduate at the University of Minnesota. So first thing to talk about is agriculture in general. Agriculture is kind of the backbone of civilization, and we really rely on our ability to grow more food each year in order to feed an ever increasing global population. Not only do we need to make more food, we also need to be constantly contending with droughts, evolving pests, and decreasing soil quality. And all of these have been getting only worse over time. Plants are amazing organisms. Over millions of years, they've been able to adapt and thrive nearly everywhere on Earth, and in some very hostile environments. Plants have the inherent ability to change, but the process of natural evolution is very slow. And while humans have been able to selectively breed plants to make them better over thousands of years, that process isn't a whole lot faster. So in order to create new and better lines of crops at a much faster much faster rate, we can employ a technique called gene editing. To kind of define some of the stuff that I was talking about first, when I'm talking about crops, easiest things to think about would be wheat, corn, rice, even tomatoes perhaps, the type of plants that we cultivate on like massive scales. Humans have a really long history of these plants, and we've come a very long way in understanding how they work and how to spot really beneficial qualities in them. In the last few decades, we've been able to track some of those beneficial qualities to where they're located in the plant's DNA. DNA can be a very complicated subject to talk about, but at its core, it's really just the instructions for how an organism grows and functions. The DNA in the seed is virtually the same as the DNA in the whole plant, and it really contains all the necessary instructions for how the plant is supposed to grow, survive, and reproduce. Those different instructions are encoded in small little strips of DNA called genes. Different genes can control different instructions that allow the plant to conserve water, grow more efficiently, or handle stress. These genes change over time. Random mutations that occur may improve plant function and then be carried on to the next generation. This process is very slow and can be sped up through selective breeding, like I mentioned. but the process of gene editing makes it much more faster and much more precise. Gene editing is essentially using techniques to tweak those instructions and improve the plant's function. There are many different techniques, but they generally do one of two things. First kind of gene editing is just making small changes to what already exists inside of the plant. This kind of editing is less radical. The changes that you make could occur through natural evolution or selective breeding just over a much longer time scale. The other kind is a little bit more radical. That's where you can take genes from completely different organisms and put them into the plant. The second kind is really cool. I did a project a couple years ago where we took some genes from fireflies and put them into petunias to make them slightly glow in the dark. But most of my work focused on the first category, which was making changes to what the plant already has going for it. Now, the biggest problem with current gene editing techniques is that they can be really expensive, they can be really time consuming, and they're also really challenging for many labs or companies to do on their own. The system that I helped to work on at the University of Minnesota aimed to make the process less expensive, faster, and more accessible to a wider range of labs and companies. Our work centered on hijacking viruses. Like people, plants have to deal with viruses too. At their core, viruses are just pieces of genetic material that go into an organism and then use their host to replicate. Some viruses really aren't all that complicated and are really just the instructions for genes in order to get in, move around in the organism and replicate. Now, we took one of the plant viruses that was known to infect tomato and tobacco, which are the two plants we were working with, and we removed some of the parts that would be very harmful for the plant and replaced them with two things. The first thing that we added into this virus was a little tag that sort of tricked the plant. Obviously, plants don't want viruses moving around in them. So when we introduced this little tag, it kind of tricks the plant into thinking, oh yes, that's something that's supposed to be there. The tag was just a sequence of DNA that was native to the plant that it recognized, that it trusted. Before we put that tag on the virus, we noticed it had a very limited range in movement around the plant. It wouldn't move much farther from the place that we injected it into the plant. But when we included the tag, we noticed it had free range to move around the plant to get everywhere we needed it to get. The second piece, which was actually involved in the gene editing, was called a guide RNA. And this was the most important piece. So as the virus moved throughout the plant, it would make copies of this guide RNA. And what the guide RNA would do is it would interact with something called Cas9. You can think of Cas9 as a pair of DNA scissors. Now, Cas9 is not found in wild plants, and the plants that we worked with had to be specifically engineered to have these scissors in them. So you don't have to worry about finding this sort of stuff out in the wild. So when the guide RNA and Cas9 find each other inside of a plant cell, the guide RNA basically guides the Cas9 to a very specific spot that we choose on the plant's DNA and makes a cut at that site. Now, DNA is constantly being broken and repaired, so plants are really good at fixing this. However, this type of cut that Cas9 makes isn't super effective at making fixes to that. So oftentimes there are a lot of mistakes that are made when it's repaired. Those mistakes make it so the end product looks different than the starting product, and it can either improve function, reduce function, or make it so a gene doesn't work at all. And by changing the guide RNA, you can essentially target any gene within the plant. And if the virus ends up making it to a part of the plant that produces seeds, those changes are then carried on to the next generation, and boom, you've made an edited plant in a single generation. The first downside to this is obviously the system is in a very early stage, and we haven't gotten it to work in important crops like corn or wheat, but it's safe to say that's a distinct possibility in the near future. And the main upside that I want to talk about is that with normal gene editing techniques, it takes upwards of a year plus to get an edit that you don't even know if it'll be beneficial to the plant. You're basically spending a year plus just to kind of test an idea that may or may not work. But with this technique, you can go from design to an inherited edit within a couple of months. It's cheaper. Again, despite how it might sound, also requires easily about 1/10 of the amount of work that's required for traditional methods. Now, as I said at the start of my talk, we are in desperate need of new ways to test new crop lines to meet the challenges of a changing world. This method, using viral mediated gene editing, is still in a really early stage, but it offers a much faster, much cheaper, and a much more accessible alternative to traditional gene editing techniques, which gives scientists a really powerful tool to engineer the plants of the future, which is something I'm really excited to see. That's all that I have. If anybody has any questions. Why did we make the petunias glow? So the project that we were working on was using luminescence, which is the process of certain organisms glowing, as a way to track what gene edits were going on. The project didn't turn out very well, so the person I was working with decided he was just going to use this as an opportunity to make a really cool anniversary gift for his girlfriend. So maybe breaking a few laws about releasing transgenic organisms into the wild, he made a glowing flower and took it home. I'll spread that one around. Why are GMOs safe? Something that I didn't talk about that adds into the process of slowing down the process of generating new crops is that there's really strict regulation that goes into place. When a company is trying to bring a new GMO crop to market, they have to go through a really strict regulatory process. Oftentimes that takes several years. They need to make sure that it's a very, there's no differences between that and your normal varieties in terms of actual nutrition or health benefits. And at the same time too, one of the important things to consider is that with most GMOs, there's nothing really there's no, glowing genes being added in there. Everything that's being changed was already in the plant to begin with. And a lot of these changes could have just occurred naturally. We're just using techniques to speed up that process. If you could have one crop that would always be edited successfully, what would it be? One plant that's really opposed to gene editing is blackberries, and I really wish there was a way to get seedless blackberries. That's something I think would be a really great project, but alas, we don't have the capabilities to do that right now. If I had to pick a second one, are you a plant hacker? Funny enough, one of the first platforms I worked on was a really long and stupid acronym that turned out to be hacker. So whenever we would talk about it, we're like, we're plant hackers. It was pretty funny. But yeah, I'm sure we can go with that title.
All right, thank you very much, Ryan. Great talk. And last but not least, we have Laurie Agosto, who is a prep scholar at the University of Missouri and who's going to tell us about some of her work studying retroviruses. Take it away, Laurie. Thank you.
Yeah, so I'm gonna start off with a small little presentation, and it specifically deals with the innate immune system. So I'll share my screen and you're able to see that, hopefully. Hold on one second. Okay, is everyone able to see that?
Yep, we can see it.
Perfect. Okay, so like Zach said, my name is Laurie Agosto. I am a second year prep scholar, and today I'll be talking to you about the innate immune system. We as humans have an immune system, but it's made up of two parts. We have the adaptive immune system and the innate immune system. So the innate immune system is a system that you are born with. So it's a broad spectrum immune response system versus an adaptive immune response is when you, when you get a vaccine, you develop memory cells. And so like they're able to attack for like in this case, like COVID or influenza, they're able to attack when you get that pathogen. But the idea is that the innate immune system, the broad spectrum immune system that you're born with, isn't as specific as an adaptive immune system. As we're finding out, it's actually more specific than we initially thought. So in the innate immune system, we have what are called pattern recognition receptors that recognize pathogen-associated molecular patterns, or PAMPs. And pathogen-associated molecular patterns are conserved regions in virus or bacteria or other pathogens. So for example, like in viruses, some pattern recognition receptors would be like the surface proteins. And we have pattern recognition receptors that can recognize those host sensors. And when it does recognize, it sends a signaling cascade to have an immune response. One of the pattern recognition receptors that I focus on is called toll-like receptors. And toll-like receptors, as I said, are a family of pattern recognition receptors. They can be located on your cell surface and inside of the cell, called the endosome, which is this pink circle here. And when they get activated, they recognize and send an immune response. And the toll-like receptors structure, I think, is really interesting. It's made up of a leucine-rich repeat, which is this little horseshoe shape here, followed by a membrane, and then a toll interleukin 1 domain. And it's the toll interleukin 1 domain that starts that immune response and signaling, sending inflammatory responses. And so all of this to set up the fact that we are looking at toll-like receptor 3, which recognizes double-stranded RNA. Double-stranded RNA can be found in influenza and respiratory syncytial virus. Now, double-stranded RNA can have different conformations. Our lab is currently studying what specific conformation activates toll-like receptor 3. And to do this, we are generating many double-stranded RNAs, and we are delivering them to a reporter cell line. And the idea is trying to figure out which conformation activates toll-like receptor 3, and if it does activate it, what genes get upregulated or downregulated? Are they the genes that control antiviral responses or inflammatory responses? And my project is very interesting because not only do I work on the wet bench and stimulating my cells and delivering these double-stranded RNAs, but I also create the bioinformatics pipeline to analyze which genes get upregulated or downregulated. And so this is called transcriptomics. So bear with me. As we, so I have the double-stranded RNAs, I deliver them to my cells and I'm stimulating them and I'm trying to figure out, okay, which one activates my toll-like receptor 3? And after I stimulate them, I collect the cells and I send them off for sequencing. And what I collect is the mRNA. So once I get the mRNA from the biocore, the bioinformatics core, so they send me back all of these RNA sequences. Now it's my job to make a pipeline to organize these sequences in a way that I'm able to, so I take the different sequences because they're cut up and I align them to the human genome. So now that I have it against the human genome, I'm able to see how many genes get upregulated and downregulated, and I'm able to visualize that using a heat map. And so if we're able to figure out what configuration activates specific genes, we can use this information to help make vaccines in the future against influenza and RSV. And this is a picture of my lab. And if you guys have any questions, please feel free to ask. Okay. So I got a couple of questions. The first question is, what is my favorite virus overall? I would have to say the one that I've learned the most about is influenza, but also in lab, we can make virus-like particles. They're not able to replicate, but the fact that we can make a virus-like particle in lab is really neat. What is the coolest thing I found so far? So there are different parts of RNAs. So yeah, there are different viruses that have RNA, and specifically the one that I'm working with right now is like poliovirus. A section of poliovirus can activate toll-like receptor 3, but that poliovirus is a single-stranded RNA. And what the single-stranded RNA does is fold in on itself, so it looks like a double-stranded RNA and activates toll-like receptor 3. So how does COVID fit into all of this? So that's a good question. So I'd be able to use the pipeline that I'm creating to identify the genes that are being upregulated and downregulated when COVID infects a cell. So I would have to look at like what toll-like receptor it's activating, and then I'd be able to use the pipeline that I've created to verify that and maybe contribute to that study. But as of right now, we're not focusing on COVID as to influenza and RSV. Do RSV vaccines tend to need to change often like the flu vaccine does? That's a good question. I'm not sure. So I would have to look a little bit more into that, but I'm not sure. And yeah, adaptive natural selection is great. Okay, so are your methods used in creating many other vaccines, or do you use different methods for other unique diseases and vaccines? So our method is like in the infancy of the lab. So we are trying out this method to see if it's a viable option to use to make vaccines in the future, but we haven't made that connection yet since we're just establishing our pipeline. I believe the current methodology that like big pharmaceuticals like Pfizer and Lilly have are well established, and I think that when our data and our research does get there, hopefully we can contribute to that. So what are my future research goals? So I'm interested in infectious diseases and host pathogen interactions. And specifically, I'm very interested in immunology from the aspect of the pathogen interacting with the host, or like the host, and seeing what defense mechanisms are there on both sides, and how does like the pathogen evade those defensive mechanisms. And so I'm applying for my PhD this upcoming cycle, and I'll be going into the programs that I'm applying to are in bioinformatics, but the ideal lab will combine my bioinformatics quantitative tools alongside with like my wet bench tools. And do all receptors look the same? No. So we have the toll-like receptor. So the toll-like receptor family, they all look the same. So they have a little horseshoe with like the little membrane, and then the the Chur domain. So those all look the same. The toll-like receptor family look the same. But then we also have like nod-like receptors, as well as which one were the other ones? Sorry. The reg-like, the reg-I-like receptors. So we do have other receptors, but they do not look exactly the same. So, yeah. if that's all the questions, thank you so much for your time.
Thank you so much, Laurie. Great talk, and best of luck in your applications. Okay, thank you everybody, to Katie, Fran, Alex, Zach, Ryan, and Laurie. You guys did an awesome job. I want to put our contact information up here in case anybody wants to reach us. Here is our Twitter, our website, and our email address. Again, we are all graduate students. We are scientists in training right now, but we are excited about sharing our work and about connecting with Missourians. If you guys want to get a hold of us, please email us or get onto our website and request an event. And we look forward to hearing from you. And thank you again to Sarah Howard and Daniel Boone Regional Library for inviting us for this event. Thank you so much.